Dear students, I am going to talk about phagocytosis process. So here I am going to talk about what is that phagocytosis process, what are the cells involved in the phagocytosis process, and then how that phagocytic cell kill the pathogen. Here, what is phagocytosis? First we should know the definition of phagocytosis. Phago means eating. Cytosis means breaking. That means here that phagocytic cell take any pathogens, for example maybe it a bacteria, virus or protozoans. It take pathogens and get digest. That digestion will be called cytosis. So phago means eating and then cytosis means breaking, taking of a pathogen or eating of pathogen. After that it will be go for digestion. That is called cytosis. So first, what are the cells involved in the phagocytosis process? Here lot of immune cells are present in our system, especially what we are going to see, how that macrophages involved in the phagocytosis process. Here the main two cells are involved in the phagocytosis process. They are called first defense mechanism cells. Number one, microphage. Number two, macrophage. So here these macrophages are called polymorphonuclear leukocytic cells. They are called dominant white blood cells. Example, neutrophil, basophil, eosinophil. So these dominant white blood cells are present in the bloodstream. So this type of phagocytic cells present in the bloodstream, that is called macrophage. Another cell which is involved in the phagocytosis process is macrophage. So these macrophages are called reticuloendothelial cells. These reticuloendothelial cells or phagocytic cells are synthesized from the bone marrow. So here that macrophage synthesized from the bone marrow, it is called that chromocyte cells. In bone marrow, the cells are called chromocytes. These chromocyte cells after differentiate or it will be released into the bloodstream, they are called monocytes. In monocytes in blood, these monocytes are having very short life period, up to 24 hours. After that 24 hours, these monocytes are transformed or migrated into the tissue. Now these monocytes once enter into the tissue, they are called macrophage. Here that macrophages are two types. So what is that two types now? One is called free macrophage, another one is called fixed macrophage. The free macrophages are located in lungs, peritoneal cavity and then lymph. Another type of macrophages is called fixed macrophages. These fixed macrophages are located in Kupffer cells. Maybe it will be called as the liver, osteoblast. So it is also called bone and lymphoid organs. And most of the connective tissues also contain these fixed macrophages. So now in our system, both blood as well as tissue having macrophage. Now next stage, how these macrophages involved in the phagocytosis process. Phagocytosis is nothing but killing of pathogens. So it will be takes place in four steps. Number one, chemotoxis. Number two, binding. Number three, injection. Number four, be called as digestion. So in phagocytosis process, first steps will be called chemotoxis. Here, what will happen now? When that pathogen enter in your system, it fix the target place. Once it will be fix the target place, or it will be reach that target place in our system, and then our system will be release some chemical factors or chemical substance. We can say simply chemical substance. They are called chemotactic factors. What are the chemotactic factors released from our system? They are called complement components. Lot of complement components are available, but here only one complement components will be released, that is called C5A. And then other chemotactic factors also released. They are called neutrophil. During that allergy condition, that neutrophils also secreted out. Another cell is mast, and then finally N-formylmethionine. So all these substances are called chemotactic factors or called chemical substance. So once these chemical substance released from our body, or that part when the pathogens plays in our system, so by the release of these chemotactic factors, the phagocytic cells get identified where the pathogens present or where the pathogens located. Once that macrophages identified the pathogen by the releasing of chemotactic factors, it will be reached nearer to the pathogens. The pathogens may be virus, bacteria, or protozoans, whatever it may be. Once they reach that pathogen, they need to get killed or they need to get attached to the pathogens. This is called the second. First process is called chemotaxis. Second process is called binding or attachment. In second stage, what will happen now? By the release of chemotactic factors, the macrophage identify the pathogens and then get attached to its surface to form pseudopodia. That means what will happen now? It will be get attached on its surface to form that pseudopodia. The pseudopodia is nothing but cover of vacuole. So it form that vacuole around the vacuole of the pathogens. After that attachment, it will be formed that vacuole around the pathogens. In second stage, once that pseudopodia will be formed, now the pathogen get vacuolarized by the membrane present on the phagocytic cells. So here, how these macrophage get attached or binding on the surface of the pathogen. So here, that phagocytic cells having that receptor on its surface, they are called toll-free receptors. So these receptors released that one protein called opsonine protein. By the release of this opsonine protein, they can get or binding or attached to the surface of the pathogens. So now the pathogen get vacuolarized and get inside into the membrane of the phagocytic cell. This is called second stage. Third stage is called injection. So once that binding or attachment is over, the third stage is called injection. So here now, after that formation of pseudopodia, the pathogen is circularized by a vacuole. So this circularized form of pathogen is called phagocytosis. So these phagocytes located in the inside of the membrane of macrophages. Once these phagocytes are formed, now the inside of the membrane of macrophages having that subcellular organelle, it's called lysosome. So these lysosome having a enzyme called lysozyme. So first these lysozymes kill the vascularized pathogens called phagocytes. Before releasing of lysozyme enzyme from lysosomes, what will happen now? That lysosomes get attached with phagocytes. Phagocytes are nothing but vascularized form of pathogen. So now this vascularized form of pathogen is called phagocytes. These phagocytes get attached or fuse with the subcellular organelle. It's called lysosome. So once this fusing or attachment is over, it is called phagolysosomes. Phagolysosome is nothing but fusing of phagosomes with lysosome. It's called phagolysosomes. So this process will be takes place in the third stage, injection. Once that formation of phagolysosomes is over, after that, that lysosome release that one enzymes called lysozymes, and then other peculiar antibacterial, antiviral substance also released in the macrophage cell membrane or inside of the cell structures. So here that lysosomes release that enzymes called lysozymes. These lysozymes kill the vascularized form of a pathogen that is called phagocytes get digested. Now that process will be called digestion. Once digestion will be over, so these breaking of pathogen, it will be exocytosis. That means it will be released from the membrane of macrophage. That process will be called exocytosis. So here, so these exocytosis over, all that breaking of pathogens will be released. So this process will be takes place in the fourth stage. It's called digestion.